my name is Sasha Woods and I'm a Postdoctoral Research Assistant at the University of Bristol.
My name is Gemma Beers and I'm a PhD student at the University of Bristol.
Today we are going to talk to you about eye and brain development, function and disease. Let's start with eye and brain development. The brain develops from a structure called the neural tube. You can think of it as being a bit like a straw. The neural tube balloons into three what we call vesicles: the hindbrain, midbrain and forebrain. Here's a diagram of what the fully developed brain looks like. The part of the brain that my work focuses on is called the hippocampus. It's this region here, located in the forebrain, that looks a bit like a bow tie. The hippocampus is important for spatial learning and memory. You know your way from your school back home thanks to the hippocampus learning and storing visual cues, like the location of a shop or a park. The eye develops from the developing forebrain, making the retina at the back of your eye a part of your central nervous system. Let's look at the structure of the eye in a bit more detail. Your eyes are required for vision. Light enters the eye through the cornea and lens at the front and is focused onto the retina at the back of your eye. The retina is made up of lots of different cell types, which are all required to process the light signals and transmit the information to the brain via the optic nerve. These cells are arranged into specific layers. The rod and cone photoreceptors are located in the outer nuclear layer. They detect the light and transmit information through messenger cells in the inner nuclear layer through to ganglion cells in the ganglion cell layer, which pass the information onto the brain. How is this information transmitted? Well, all the cell types mentioned here are called neurons. Neurons pass messages to each other via their branches, or what we call processes. Dendrites carry information in, and axons carry information out of a neuron. So how do the cell types discussed here and all other cells of the body know how to develop properly? The instructions for a cell's development is encoded in a special molecule called DNA. DNA codes for proteins, and proteins are the building blocks of cells, making different cells good for different jobs. Sometimes there is a mistake in the information encoded in DNA, and we call this a mutation. Part of my work involves looking at mutations in specific genes and the effect that it has on the structure and function of eyes and brains, and the other genes expressed within them. You can see that mutation in a specific gene causes the retina to be much thinner due to a loss of cells in this layer. A mutation in this gene also causes a loss of cells in the hippocampus, affecting spatial learning and memory.
Most of the time, our bodies develop properly, but sometimes things go wrong. If germs get into our body, they can cause infections and disease, and it is the job of our immune system to protect us. Our immune system is made up of many kinds of white blood cells, and phagocytes are the first types of white blood cells that help us when our body is under attack. The word phagocyte means eating cell, and this is because their job is to swallow up the germs and destroy them. Phagocytes can tell what is good and what is bad by checking for patterns. And because the patterns on germs look different to those found on healthy cells in our body, it knows which ones it needs to swallow. Once the phagocytes have gobbled up the dangerous germs, they produce powerful chemicals which attract lots more white blood cells to help in the fight. When you get a cut, you might have noticed it sometimes becomes red and swollen, maybe hot and sore. This is called inflammation, and it happens as a result of all the white blood cells fighting off the germs. Inflammation is your body's way of protecting us, but sometimes phagocytes need some extra help to keep the germs away. T helper cells are a very special kind of white blood cell, which help the phagocytes when it cannot kill the germs on its own. The phagocytes chew up the germs into tiny pieces and show them to the T helper cells, which produce lots of chemicals to kill it. It is really important that our immune system can tell the difference between germs and normal cells, because if T helper cells in the blood mistake cells in our body for danger signals, they can become activated and use their killing powers to attack our organs. This is known as autoimmunity. Auto means yourself, and immunity refers to your immune system. If this happens to cells of the eye, you can get damage in the retina, which can cause blindness. We can use slides and a microscope to learn more about what happens during eye disease. First, we take very thin slices of the eye, which has been frozen in a kind of wax, and then use chemicals to stain the cells different colours. Here you can see a picture of a normal eye. The retina is shown in purple, and you can see the layers are very straight. In a diseased eye, lots of white blood cells flow into the eye, which are the brown cells you can see here. They produce lots of damaging chemicals and cause the layers of the retina to become disrupted, which can damage your sight.
Maybe as a result of watching this vlog, you'd like to do your own research into genes that are important in eye and brain development.
Or maybe you'd like to do some research into other autoimmune diseases and the organs that they affect.
Thanks for watching.